Welcome to this episode of Neurology Video Journal Club. My name is Payam Mohassel. I'm a neuromuscular neurologist and associate professor of neurology at Johns Hopkins University. And today I'm joined by Dr. Andrew Mammen, and we'll be discussing a paper by Galea et al. Published in 2022, which looks at the role of interferon type 1 on muscle stem cell proliferation in dermatomyositis. Andy?
Yeah, so I'm Andy Mammen, and I am a senior investigator and leader of the muscle disease section at the National Institutes of Health. And like Payam, I'm really interested in muscle diseases and especially autoimmune muscle disease. So I think I bring a little bit of expertise about autoimmune muscle diseases, and Payam is a real expert on muscle regeneration. So hopefully between the two of us, we can help guide you through this paper. So I wanted to start just by giving a little bit of background on autoimmune muscle diseases. They're pretty rare diseases. It's a heterogeneous family of diseases. And when I think about myositis, I think about the four major types. That would include dermatomyositis, which we're going to talk about today, but also the anti-synthetase syndrome, immune-mediated necrotizing myopathy, and of course, inclusion body myositis. Even in patients who have dermatomyositis, we really think of it as being a heterogeneous disease. And the different types of dermatomyositis can be defined by the different myositis-specific autoantibodies that we find in these patients. So the major ones are autoantibodies that recognize TIF1 gamma, MI2, NXP2, SAE, and also MDA5. And so we'll talk about some of those autoantibodies because they're relevant to the paper that we're gonna talk about today. Now, dermatomyositis, of course, doesn't just involve muscle; it involves skin and sometimes lung as well. But and we don't really understand the disease pathogenesis very well. But one thing that's been known for more than 20 years is that type 1 interferons seem to play a prominent role in the disease. So more than 20 years ago, Steve Greenberg and his colleagues found that if you looked at muscle biopsies from patients with dermatomyositis, you could find massive upregulation of genes that are known to be induced by type 1 interferons. And since then, it's also been shown that there's what's now called a type 1 interferon gene signature in blood cells as well as skin in dermatomyositis patients, and that the degree of upregulation of these genes that are part of the interferon gene signature correlates with disease activity. So we've known from those experiments type 1 interferons are important, and now we have a class of drugs that are pretty good at inhibiting the type 1 pathway, and that would be the JAK-STAT inhibitors that are used a lot in rheumatology, but not so much in neurology so far. But we'll get more to that later. In terms of the muscle biopsy features of patients with dermatomyositis, a sort of classic feature is perifascicular atrophy, so relatively normal looking muscle fibers within the center of the fascicles, but a lot of small, atrophic, degenerating and regenerating muscle fibers around the periphery. So that's kind of my background on dermatomyositis and autoimmune muscle disease. I'm going to turn over to Payam. Maybe you can tell us a little bit about how muscle regenerates when it's damaged.
Yeah, I mean, I think actually one of the reasons I got interested in studying muscle was its capacity to regenerate. So muscle maintains this ability to regenerate itself after injury, and this becomes kind of a very curious and important thing to consider in disease states, because regardless of what the underlying cause of the injury to the muscle is, whether that's from autoimmune disease or genetic disease, we still have this potential for regeneration, which is mediated through these adult stem cell satellite cells that reside in the muscle tissue. So normally these satellite cells or adult muscle stem cells are quiescent. They're just sitting there within the muscle. But when the muscle gets injured or through different signaling, even some weight bearing or minor trauma can sort of start them up to start to undergo changes that result in them proliferating first. So they make a lot of these myogenic progenitor cells, which then go down the pathway of differentiation into and a programmed which then are programmed to fuse and sort of repair the muscle that was damaged. Now, it's really interesting that as I mentioned in the diseases, we still have these satellite cells, but these satellite cells can't quite keep up with repairing the muscle adequately. And there's been a lot of interesting research over the past couple of decades looking at this. And as it turns out, satellite cells are actually not a homogeneous group of cells, and there's different subpopulation of them. So there are some that are a subgroup of these satellite cells that are more likely to kind of regenerate and undergo divisions and replete the pool of satellite cells. And there are ones that are further down the path of differentiation and undergo this asymmetric division, make a lot of progenitor cells when the injury occurs. And there needs to be a balance between these two modes of division. If you have too much symmetric division, so you're only making satellite cells and you're not making cells that are going to help repair the muscle. If you have too much asymmetric division, you're sort of depleting your satellite cell pool over time. And it turns out that there's different diseases that it's been now sort of figured out that they have those defects in their satellite cell behaviors. For example, it's Duchenne muscular dystrophy has been studied extensively in this, and it's clear that in that disease, it turns out that the symmetric division is going on far too much, and there's not enough asymmetric division of satellite cells. On the flip side, in aging or some age-related diseases, even IBM, inclusion body myositis, there may be more asymmetric division going on and not enough symmetric division. And so it becomes important if one wants to think about how to intervene to sort of correct these imbalances of satellite cell behavior as a therapy for different diseases. So with that, we can actually, with that background, we can sort of start delving into the paper, and I'll hand it off to Andy to give an overall picture.
Yeah, I think this paper is really interesting because it sort of combines these two things that we've been talking about so far. One is dermatomyositis and the role of type 1 interferon, and the other is muscle regeneration and the balance there. So this is a really nice study by Laura Gallet and her colleagues, and this was done to really try to understand the potential role of type 1 interferons in modulating muscle regeneration. In particular, they focus on muscle stem cell proliferation. And what we're going to talk about is how they isolated stem cells from dermatomyositis patients and healthy controls and evaluated their ability to proliferate, to differentiate, and also their the increased capacity or I should say decreased capacity of proliferation in dermatomyositis stem cells. And then they looked at whether this might be mediated by type 1 interferon, and they're actually able to show that type 1 interferon that seems to be secreted from the myoblasts themselves has an autocrine effect on muscle cells that decreases their ability to proliferate. So with that sort of summary of what's ahead, I'll have Payam explain what's going on in our first slide.
In the first table, which is not shown here, in the first figure of this paper, they're basically telling us how they what the samples were that they were dealing with. So they had 8 dermatomyositis patients, and these patients were diagnosed based on the most recent ENMC criteria. And then they looked at the muscle biopsies of these 8 patients, and they categorized them to a mild dermatomyositis group over here, and a severe dermatomyositis group based on the histological features. And they used these histological criteria that have been published several years ago now, originally developed for juvenile dermatomyositis, but they applied it to the adult dermatomyositis patients that they had here. So as you can see, for example, in this patient with mild dermatomyositis, there's some perivesicular atrophy, but in the severe case, there's a lot more inflammation and perivesicular atrophy in the myofibers in the perivesicular regions. Likewise, looking at CD56, which is a marker of regeneration, or NCAM is the other term for it, you can see there's a lot more regeneration going on in the perivesicular region. or there's a lot more NCAM positive fibers, I should say, in the perivesicular region in this severe case compared to the mild dermatomyositis. And another feature of dermatomyositis is this vascular dropout. So the blood vessels are damaged as part of the disease. And here you can see a marker of endothelial cell CD31, and you can see there's some areas of endothelial cell dropout in this mild case, whereas in the severe dermatomyositis case, histologically, there's a lot more capillary dropout. So they used these and other histological criteria to divide up the patients into the severe and mild based on the way the muscle looked. And then they took this further to study them in more detail.
Yeah, and so in the next slide here, we are still looking at muscle biopsy specimens from healthy controls and patients with a severe histological form of dermatomyositis. And what they're staining for here are PAC7 cells. So these are the muscle stem cell precursors that are present both in healthy control muscle and in the muscle from patients who have more severe dermatomyositis. And what they did is they're trying to quantify the number of these stem cells that are present in healthy controls and dermatomyositis. And they're also very interested in looking at the perifascicular area, where we think most of the damage is, versus the intrafascicular area. And what they found is that in severe dermatomyositis, there are many more PAC7 positive stem cells than there are in healthy controls. And this really seemed to be restricted to the perifascicular area, because if you look in the intrafascicular regions, there are pretty much the same number of PAC7 positive satellite cells in both healthy controls and even in the severe dermatomyositis muscle biopsies they looked at. So here we're seeing, evidence of muscle regeneration, and there being more myoblasts, or early stage regenerating muscle fibers in the dermatomyositis than in the healthy controls. And again, restricted to the perifascicular region.
Great. They then took the same muscle biopsy samples that they had of these patients, and they went ahead and isolated these muscle stem cells, the satellite cells, from them. And that requires sort of digesting the muscle tissue and then purifying those cells based on markers. And they used NCAM as a marker of these cells. And after isolating these muscle stem cells, what they abbreviated to MUSCs, when you grow these cells in culture, depending on what sort of additional things you add, such as insulin and removing serum that's there, you can actually walk them through this program of first proliferating into myoblasts and then differentiating into more myocyte-like cells, and then actually fusing into myotubes. And you can actually see that in the culture dish and evaluate the efficiency of these different cells from different patients, from healthy controls versus dermatomyositis patients, in these different stages of proliferation and differentiation. So there are ways to look at the efficiency of proliferation, and the EDU assay is one that's commonly used to look at how well can these cells proliferate, divide. And when they did that, they found a significant reduction of proliferation of dermatomyositis patient-derived muscle stem cells. Likewise, when they looked at the differentiation, this was also significantly reduced in efficiency. For differentiation, they were looking at myogenin, which is a marker of cells that have sort of moved away from being stem cells and more being committed to muscle-like cells in culture. They also looked at fusion, looking at fusion index, which is again another way of looking how well the cells are forming myotubes, and again found a significant reduction overall in this. Now, because proliferation was reduced, it's possible that the lack of proliferation was affecting the other steps down this path of differentiation and fusion. So they really focused their attention from then on the proliferation part, although it's possible that in dermatomyositis, there may be also an additional defect in these additional steps. They also had a few samples from juvenile dermatomyositis patients, and they found the same reduction in proliferation efficiency in those muscle stem cells.
All right. So in the next set of experiments, they were really interested to see whether type 1 interferon might be contributing to the decreased ability of these dermatomyositis muscle stem cells to proliferate. So the first thing they did was they took healthy control muscle cells and they grew them either in the absence of type 1 interferon or in the presence of increasingly higher concentrations of type 1 interferon. And what they showed is at relatively low concentrations, there was actually an enhancement of the ability of these myoblasts to proliferate. However, at the higher concentrations, there is a decreased ability of the cells to proliferate. I'd point out here, I don't think we really know what the concentration of type 1 interferons in the muscle is. So we don't really know whether they're experiencing something that's more like the low levels of interferon that promote proliferation or the higher levels that inhibit it. The next thing that they did was to take healthy control muscle cells and grow them in the presence of media that had been conditioned by patient myoblasts from dermatomyositis patients. And what they found is if you take the media that had been conditioned by the myoblasts that were from severe dermatomyositis patients, you actually recapitulated the effect of type 1 interferon. That is, you'd see a reduction in the ability of the cells to proliferate. So they suspected, hey, maybe this is mediated by type 1 interferon. I'd point out there in B, there does seem to be a typo on the x-axis. You can see at the very left, it's healthy controls, and the next is the severe dermatomyositis. I think that next one should probably be moderate dermatomyositis, but instead it's labeled healthy control. Again, I think that's just a typo. Now, in the panel C there, what you can see is they've taken myoblasts from patients with severe dermatomyositis, and you can see in the left there, there's a certain level of proliferation, which we think is going to be low. But if they add to the medium a JAK-STAT inhibitor, ruxolitinib, you get enhanced proliferation. And you also get enhanced proliferation if you add to the media a interferon alpha receptor blocker. So both of those things really suggest that it is interferon that is inhibiting the proliferation of the myoblasts from the patients with severe dermatomyositis. In the next panel, they've taken healthy control muscle stem cells and add supernatants from severe dermatomyositis muscle myoblasts that have either been treated with ruxolitinib or not. And if you don't treat the severe myoblasts with a JAK-STAT inhibitor, you just take that media and put it on the healthy control cells, there's inhibited proliferation. However, if you get that conditioned media from dermatomyositis myoblasts that have been treated with a JAK-STAT inhibitor, which should decrease the production of interferon into the media, then you don't see that effect. So that also is supporting evidence that this really is a type 1 interferon-mediated effect. I think, I'd point out one thing I think they could have done to improve the study was to show that if they add a neutralizing type 1 interferon antibody, that they could also block the effect of the supernatant from the severely affected dermatomyositis myoblasts. But I think that it does pretty convincingly show that there's an autocrine effect and that it is mediated by type 1 interferon, and that's what is contributing to decreased proliferation of the cells from those patients.
So, overall, I think, this paper adds quite a bit to what's already known about interferon type 1 and dermatomyositis. In particular, they clearly show this autocrine effect of interferon type 1. And so the cells that are derived from dermatomyositis, if you even just take the media that they make and then put it on the healthy control cells, you introduce those effects, which shows that point very very nicely. And this effect is clearly on the proliferation, and it could also have an effect on differentiation of those cells. And I think another sort of important takeaway from this is that this may actually explain why some dermatomyositis patients are so refractory to treatment, even though you've sort of treated the autoimmune effects. If you're not addressing this autocrine and potentially feed forward loop, and muscle regeneration is not has not recovered to the normal levels, that may explain why some of the patients sort of never recover to full strength even when they're treated. And so I think it adds a lot to that explanation that we encounter in clinic.
Yeah, I think I totally agree with those points. I think I would point there are a couple limitations of the study that we'd be remiss not to point out. One is it's a pretty small sample size, so only 8 adult myositis patients. And also, as I mentioned at the beginning, there are different myositis-specific autoantibodies that define different types of dermatomyositis. And only 2 of those are represented in this study. So half of them, 4 of the samples are from patients who have anti-MDA5 autoantibodies, and 2 are from anti-MI2 positive dermatomyositis patients. And interestingly to me, I usually think of the anti-MDA5 dermatomyositis as being relatively hypomyopathic, if not amyopathic. So half of those patients really have minimal muscle disease at all. And yet, at least in this study, there was still defects in muscle proliferation that they could see in those myoblasts derived from those patients. Another thing I don't know if it's a limitation so much as a question that remains in my mind about exactly how to interpret this study. The question is the decreased proliferation that we see all due to extrinsic factors, or is there some intrinsic change that occurs in the myoblasts that keeps them in this state of low proliferation and maybe making interferon? I'd point out that when they derive these cells from patients, they actually freeze them down. So they're not seeing type 1 interferon, for a certain period of time, and yet they continue, when you thaw them and bring them out and proliferate them, they continue to have this decreased proliferative capacity. So exactly how that mechanism works, I'm not sure, whether it's epigenetic or whether there's some other mechanism that changes the intrinsic properties of those myoblasts. But what do you think about the sort of clinical implications and future ways to go with this stuff, Payam?
Yeah, I mean, those are excellent points. I think I think this study is really sort of thought-provoking and opens up a lot of questions to pursue in the future. I think the more immediate one, as far as translational, is that this adds to the body of literature that's out there about type 1 interferon and potentially intervening in that pathway as being beneficial for dermatomyositis. This issue of satellite cell heterogeneity and the different types of division of satellite cell symmetric versus asymmetric in different disease states is also really, really intriguing. And of course, it wasn't explored in this sort of pioneering study, but I think it would be really interesting to tease that out a little bit in the future to see whether in dermatomyositis there's clear signature of one type of abnormality in satellite cell division. And if that's the case, then the treatments may then be geared toward that. As a side note, JAK-STAT inhibitors are known to kind of promote more symmetric division than asymmetric division. So, and it seems like, based on the data in this paper, that dermatomyositis satellite cell may be more prone to have more asymmetric division, although it's not fully clear. So that would also add another justification and rationale to use JAK-STAT inhibitors to use in this. This would be, for example, opposite of what's happening in Duchenne muscular dystrophy, where there's more symmetric division. So I think understanding what's really happening with the satellite cells may actually also inform what type of therapies we may choose to treat these different diseases that address this issue of satellite cell division. So overall, I think another question maybe I had for you, Andy, that maybe you can talk about is, what are your thoughts? You kind of briefly mentioned this about the antibodies, but do you think it's possible that some autoantibodies, some dermatomyositis patients with different autoantibodies also may have actually really different pathways involved in their muscle tissue, and therefore, understanding those more specifically, subcategories of patients also can inform. Maybe, you can talk about that.
Yeah, I think that that's a really good point. We do know from some of the work that we've done that the transcriptional profiles of dermatomyositis patients can vary depending on what their myositis-specific autoantibody is. So it very well could be that there are different pathophysiological mechanisms. And so I think that's a super interesting area to pursue further. I would just emphasize, as we wrap up, it's been known now for 20 some years that type 1 interferons are important. And I think for me, this is really one of the first studies that has helped explain how they might play a pathophysiologic role, that perhaps that they are inhibiting proper muscle regeneration, and that's contributing to the disease. They may have other effects as well, and it wouldn't surprise me. But I applaud this paper for sort of closing the loop a little bit, at least on that aspect of dermatomyositis disease pathogenesis. So thanks, Payam, for joining me to talk about this. I really appreciate your expertise and insight into this work.
No, thank you. It was great.